my next assignment, which I assigned to myself, was to talk about some issues that arise around the whole editing business. And one of the first things I'm going to talk about, and Jennifer introduced this very briefly, is the fact that there are alternatives to the standard Streptococcus pyogenes Cas9. And one of the first ones to be appropriated was the Staph aureus Cas9. This was promoted by Anne Ran in Feng Zhang's lab. And one of the reasons that they got interested in this is because the pyogenes Cas9 is a big protein, 1,358 amino acids. And it's a little, 1,368 amino acids. It's a little cumbersome to work with in some situations. And the Staph aureus Cas9 is only 1,053 amino acids, 300 amino acids shorter. People have also done a little bit of work with this Streptococcus thermophilus Cas9, which is a little bit bigger, 70 or so amino acids bigger than Staph aureus. But the Zhang lab isolated CRISPR RNAs and tracer RNAs, made themselves single guide RNA in the way that the Doudna and Charpentier labs had made a single guide RNA for the strep pyogenes system. They found that you needed a guide length that's a little bit bigger. The pyogenes system works quite well with a 20 nucleotide guide sequence. The Staph aureus system likes something that's a little bit longer, the guide sequence itself a little bit longer, attached to this framework. Recently, Jin-Soo Kim's group began working with an even smaller Cas9 protein. This is from Campylobacter jejuni, and it's only 984 amino acids. The coding sequence for it is under 3 kb. And what they showed was that they could make a single guide RNA using the same sort of principles as for the predecessors, and then they looked at guide lengths. And sort of similarly to the Staph aureus system, guides of 21, 22, 23 nucleotides seem to work a little bit better than the 20 nucleotide guide sequence, just this sequence that directs the Cas9 protein to its target. They also showed that the jejuni Cas9 is quite specific, and it can be used in these mouse myotubes and in by injection into mouse muscle. And I'll talk about this again on Wednesday, but one of the reasons for looking for these effective shorter Cas9s is that a popular vector for gene delivery in human cells and in other mammalian cells is the adeno-associated virus. And they're vectors based on AAV, but they have a limited capacity. And if your Cas9 takes up less of that capacity, there's plenty of room for a guide RNA sequence and a promoter driving its expression, a promoter driving expression of Cas9, and then some other bits and pieces, all within the packaging limit of adeno-associated virus. And then these are adeno-associated virus delivery to cells in culture and to living muscle. Okay? Jennifer showed a later version of this slide. Mine's from a review. Eugene Koonin gets a lot of mileage out of when more sequences are accumulated, they make more sequence comparisons and they expand the list of these CRISPR systems. But even as of 2015, they had identified this type 5 within class 2, where there are Cas9-like proteins that are called CPF1. And CPF1, Jennifer didn't have time to go over it, although she showed you a slide that illustrated it. This CPF1 has a bunch of differences from the Cas9 system, which may be useful for some purposes. First of all, there's no tracer RNA. There's a single RNA that's involved, so you don't have to construct a single guide RNA out of CRISPR and tracer sequences. There's just one RNA. In all of the Cas9 systems, it's the 5' end of the guide RNA where the guide sequence lives and that recognizes the target. In the CPF1, it's the 3' end of the guide RNA that recognizes the target. Instead of making blunt cuts, it makes staggered cuts that leave a 4' 5' overhang, just like a lot of restriction enzymes. And people have considered that this might have some advantages, although I don't know if anybody has taken advantage of those possible advantages. The PAMs for the CPF1s that I know about are all AT-rich rather than GC-rich, which has been true for some of the Cas9s, and the CPF1s have been shown to have quite high specificity. So this is just some more stuff. This is again from Feng Zhang's lab, looking at guide lengths, and you can see anything from 18 to 22 is an effective guide for in vitro cleavage. There's very little tolerance of mismatches as you get close to the 3' end of the guide sequence. Sorry, toward the PAM proximal or 5' end of the guide sequence itself. And they also demonstrated activity in cells, CPF1s from 2 different bacteria compared to the strep pyogenes Cas9. So this is a very functional system that people have used. One of the things that people thought might limit the utility of the original Cas9 system was the fact that you had to have a pair of Gs in the genomic sequence next to where the guide would bind. So your target had to be defined by a pair of Gs. But now, with some of these other natural proteins, the CPF1 proteins that have these AT-rich PAM sequences, the Staph aureus and Campylobacter jejuni have somewhat different guide sequences. I hope everybody's familiar with the nomenclature that R means purine and Y means pyrimidine. So, for example, this jejuni Cas9 recognizes A or G, T or C, then A, C, 4 base pairs away from the sequence that is represented in the guide sequence of the guide RNA. Okay? And then Keith Jung's lab has gone about deriving Cas9s from both Staph aureus and strep pyogenes that recognize somewhat different sequences. Instead of GG, there's one that recognizes GAG and another one that recognizes GCG. And they made one from Staph aureus that doesn't care about that G, but only cares about purine T. So, and this is by in vitro selection. So now you have a variety of PAM sequences, so you're less limited in the genomic targets you can access. Another thing that people have done with Cas9, mostly to improve its specificity, is to break it into 2 pieces. And you can break it into an N-terminal half and a C-terminal half and express them separately. The Doudna lab actually did this in a somewhat more intentional way than just cutting it N and C terminal. But you can set it up so that one of the so that the association between the N and the C terminus doesn't happen except in the presence of a small molecule or some other initiating condition. This is one, again, this is from the Zhang lab, where rapamycin encourages the dimerization or the association of the N and C terminal halves, which then go to the nucleus and have activity. Now, the activity, the inherent activity is somewhat compromised compared to the wild type intact protein, but the specificity is actually quite good because there's nothing going on until you introduce the rapamycin. And I just gave you a list of some of the other ones. Luke was involved in one of these, and there's even a photoinducible dimerization system that has been developed. So you can look those up on your own. And one of the things that people tend to get freaked out about in all of these editing technologies is what's happening at other places in the genome. What are the off-target effects? And the concern about this was stimulated and enhanced by some papers that were published in 2013, just a little over a year after the original Martin Jinek, Doudna-Charpentier publication showing what you needed in order to operate the CRISPR system. This is from Keith Jung's lab. So they have a situation where they've got a single copy GFP gene in cells, and then they're introducing different guide RNAs that will cut the GFP and by non-homologous end joining ultimately disrupt the GFP coding sequence. And they're just looking by fluorescent activated analysis what proportion of cells are now GFP minus. Okay, so it's just a disruption assay. So they've got 3 different guide RNAs that have different GC contents that are pretty good when there's a perfect match between the target in the GFP gene and the guide sequence. But then as you begin introducing single base changes, you find that if those single base changes are near the 5' end of the guide RNA, they have essentially no influence on this or this guide RNA and its effectiveness, some greater influence on this one. And even when you begin inducing pairs, either adjacent pairs or more distant pairs of mismatches, you're still getting kind of a lot of cutting with some of these guide RNAs. Then they went to endogenous genes to look to see what's going on. So this is the target. And in the genome of these human cells, there are 3 different types of human cells, there are some sequences that are related but not identical to the intended target. So let's look at U2OS cells. They're getting 50% mutagenesis in U2OS cells at the design target. But they're getting a similar, almost identical level of mutagenesis at a target that differs by 3 base pairs. And here's one that differs by 4 that's giving an even higher level of mutagenesis. So, you know, are we shooting ourselves in the foot or some other more vital organ when we are operating this system? This is from the Feng Zhang lab showing a similar thing. It's a little difficult to assimilate this right away, but what they're showing, when there's deep blue, that means a single base change doesn't interfere with cleavage, okay? And they're using similarly, they're using a system where they've got guide RNAs that are mismatched at a single location to a genomic target. So the single base changes near the 5' end of the guide RNA are not inhibiting cleavage very much at all, although when you get closer to the PAM, then it makes a pretty big difference. And this is again from the Zhang lab. If you put in more Cas9 and guide RNA, this is using a plasmid delivery system, you get more indel formation at the design target, but you also get more at off-target sites. And if you compare, you make a specificity calculation, design target over secondary target, the specificity goes down as you put in more of the nuclease.
So I assume that people have shown that preference for the amino acids near the, base pairs near the PAM need to be stronger is also mirrored in CPF1 where the 3' and 5' are flipped? Yes, but
CPF1 actually is less tolerant of single base changes at either end than the Cas9 system. But it is true, I think, that it's at one end, and it might have been in a previous slide, and I just don't remember right now how it goes. You can look it up. So if the harder you try to hit your target, the more you're going to hit the secondary targets as well. So My view, all the way from the time those papers came out, a little less than 4 years ago, has been the CRISPR system actually needs to have room for mismatches. And the reason for that is that when a virus infects the cell, even if this cell has incorporated a sequence from a relative of this virus and now is protected against that relative, the infecting virus this time may not have an identical genomic sequence. Viral sequences evolve at a really high rate. And so, you know, even keeping coding sequences the same, you can have redundant codons that would have sequence changes. And so if you were so specific that even a single base pair mismatch between the CRISPR RNA that was generated from this spacer, single mismatch between this and the incoming virus, if that did away with your immunity, you'd be in big trouble. So mismatch tolerance, I think, is adaptive in the CRISPR system, and we should expect it to have less than perfect specificity. Okay, so how much we're going to talk about how much we care, how we measure some of the off-target influences, and what we do about it. Okay? Now, how much do we care? Well, it depends what you're doing. If you're working in a model organism and you're making new mutants and you're going to study gene function through this mutagenesis, you can make independent mutations in the target and study them to see if they have the same phenotypes. You can clean up the background in a lot of genetic organisms by outcrossing, and you can complement your mutant with a wild-type gene to make sure that the phenotype you're studying is due to that mutation and not to something that happened elsewhere in the genome. If you're working with food organisms, you're concerned about safety, but in both livestock and in plants, you typically go through a very narrow bottleneck. You're essentially cloning the organisms and then expanding them through sperm donation or seed propagation. And so you can do whole genome sequencing and look to see what else did we hit when we were trying to hit gene X, okay? Now when you go to clinical applications, again, we're worried about safety. And when people talk more specifically about some of these clinical applications, you can see that in somatic therapies, particularly ex vivo modifications of cells that are then going to be put back into a patient, you typically have a limited range of off-target modifications that might come up and bite you. So as long as you kind of have those covered, you're probably in pretty good shape. As you get to stem cell therapy, somebody asked the question earlier about stem cells. Right now, I'm more concerned about the stem cells than I am about the CRISPR-based modifications of them, but that'll change over time. In germline therapies, as Jennifer highlighted, there are lots of reasons to be concerned because you can't go backwards. You're influencing an individual and that individual's ultimate offspring, and you could conceivably try to go back and reverse the change using the genome editing technologies again, but if there are multiple things happening off target, that could be a problem. So how do we assess what's going on at secondary targets? And there are a bunch of ways to do this, and I'm going to illustrate a few of them. One thing that people will do is to either make predictions just based on sequence similarities. I think Jennifer Listgarten will show you that you can do something beyond just looking at sequence similarity. Or you can use an in vitro preference assay, binding preference assay, something like CLEX. And then there are a bunch of things. This is another in vitro method that David Liu's lab came up with. But there are a bunch of things you can do that are actually assessing what's going on in cells. And one of these that was developed in Keith Jung's lab is GuideSeq. Now, if you read Keith Jung's papers, you'll know that he develops a lot of very, very useful tools in this area, but none of them is developed without a cool acronym. I think they probably are developing other technologies in their lab, but they discard them if they can't think of a cool acronym. So this is GuideSeq, and you have to go to the paper to see what GUIDE stands for, because I don't remember. But it's actually a neat technology. So what they reasoned was, when you make a break, the ends will often go back together, and we even know that you can get an end from one break to go together with an end from another break. Well, what if we put in a little piece of double-stranded DNA, our double-stranded DNA, and we just look for end capture of this double-strand DNA by non-homologous end joining? And if there's enough of that around in the cell, instead of the two ends at the break coming back together with each other, they'll capture this oligo in between. So this is a 34 base pair, double-stranded oligonucleotide. It has 5' phosphates, which help with ligation, but the ends are protected chemically from degradation. And it doesn't work well unless you protect those ends. So you're getting blunt joins for the most part. Now, this is in the genome only at sites where a break was made. And so you can use this as a sort of a landing pad for PCR and amplify the sequences that are beside where that oligo got incorporated in the genome. And this is just an elaboration of what you do to get this ready for DNA sequencing. But when a break has been made, what you should do is you should read out of this oligo tag, it can go in either orientation, read out of that oligo sequence into genomic sequence in both directions away from the place where the double-strand break was made. And so this is a way of looking to see where in the genome are there double-strand breaks. And the data from an experiment like this looks like that. And there will be a quiz on this. So what I'll do is I'll just show you one example. Not the best and not the worst example. Now, this is a site in the VEGF-A gene, and there's the target sequence up there. And then these are the number of sequence reads from a GuideSeq experiment for the intended target, which is that black square, and a bunch of other genomic sequences. Okay? And the horrifying thing from this is that the intended target is only number 3 in this list. There are 2 others that have 3, this has 2 and this has 3 mismatches. That line right there shows that any nucleotide is acceptable between the PAM and the guide sequence, okay? And that was expected. But these guys are being captured more frequently by this experiment than the intended target, which represents only 11% of all the reads. So this again is suggesting that there's a lot of off-target cleavage going on. Another procedure that's been used, this has been adopted by the Zhang lab, although it was invented by these characters. And this is one where whereas the GuideSeq is cumulative, so the oligo is there, the cleavage reagent is there over a period of time, and any break that's made during that period of time is a candidate for picking up the oligo. This is one where the only thing that's gone into cells is the cleavage reagent. Then you fix the cells and you capture whatever breaks, whatever the standing level of breaks is at that time, you capture those breaks with, by ligation to an oligonucleotide. It's a hairpin oligonucleotide. I think it's this one. You capture it with a hairpin oligonucleotide, then you shear or cut again, you capture these things with magnetic beads, and then you ligate something onto the other end. So you've captured, with this initial oligo, you've captured an end that was made by the cleavage reagent in cells. And the Zhang lab has used this, and these are just data from the paper where they did that, where they're showing levels of sequence reads from that experiment at the design target and some secondary targets. And one of the things they were trying to demonstrate in this paper is that for these particular targets, the Pyogenes Cas9 seem to cut secondary sites more frequently than the Staph aureus Cas9. But that was a way of capturing sites that had been cut by either of these Cas9 proteins. Okay? Capture them in that snapshot. This is a system that was developed in Fred Alt's lab for a different purpose, but then repurposed for this. And I'm not going to go through the whole thing, but what they're basically doing is they're using the design target kind of as a guide-seq oligo to capture cuts that happen at other sites in the genome. Okay? So if you make 2 cuts in the genome, as I told you, you can get these translocations, right? One cut finding the other cut and getting a translocation. Well, in fact, that's what they're depending on. They're depending on these translocations. So here's an example where the intended break was made in the the RAG1 gene on chromosome 11. And then when they pulled out everything that was connected to the break at RAG1, they found that there were sequences from elsewhere in the genome, and that's what these little fountains are showing. If they're red, they're pretty common translocations. If they're just sort of beige or sort of yellowish, then they're less common translocations, but to other places around the genome. And so that's identifying secondary sites that have been cut while the primary site was also being cut. This is a procedure that was developed in Jinsu Kim's lab in Korea. What they do is they identify targets for a particular Cas9 guide RNA complex by in vitro cleavage. And they drive this really hard. So take total cellular DNA, cut it with Cas9 and a guide RNA, and then just randomly sequence all the molecules in the genome. And for any site that's susceptible to cleavage, you shouldn't get any reads that cross that cut site. Okay? So they drive it hard, they pick up the intended site, and they pick up secondary sites. Then you take DNA from cells that have been treated with that guide RNA and Cas9 protein, and you should generate indels in some of the copies of those sites, and now the sequence reads will go across instead of all stopping at the break site. And so you use that to identify targets that Cas9 can cut and apparently does cut and mutate in cells. Okay? So there are all these procedures. Even before GuideSeq, people had used a capture of a viral genome, an integration defective lentiviral genome. It works by the same principle, somewhat less effective. And each of these procedures has some drawbacks. They all seem to be pretty good at identifying secondary sites, but as I said, GuideSeq, and it's true of this IDLV capture, they're cumulative. So you may overemphasize secondary targets as the cells grow and you continue cutting. BLESS is just a snapshot. It's very dependent on the timing of the snapshot you take relative to the introduction of the nuclease. DiGenome-seq requires very deep sequencing, and this HTGTS, high throughput genome translocation sequencing, that depends on getting at least 2 breaks in the cell, so you have to kind of overcut in order to see anything. So they all have some drawbacks, and someone should sit down and compare these with dozens and dozens of guide RNAs to see if they all agree. Okay. As an alternative to worrying about and measuring these off-target effects, people have also begun working on improving the specificity of the nucleases so that you have less reason to worry about off-target cleavage. And one of the early ways of doing this was to turn Cas9 into a nicking enzyme. Jennifer said that with these anti-CRISPR proteins that interfere with the access of the H&H domain to DNA, that you actually inhibit cutting of both strands. But if all you do is you make a single amino acid change in the protein so that the H&H domain, for example, is not cut, the other active site can cut the guide strand, and it can cut the guide strand wherever in the genome Cas9 has to bind. So if you provide two guide RNAs targeted to opposite strands, pretty close to each other, the cell looks at this as if it was a double-strand break, a frank double-strand break. But what you've done is you've squared the specificity of the system, because now both of these guide RNAs have to bind in order to make the nick. So this improves specificity, and remarkably, these guide RNAs can bind to sequences that are up to 100 base pairs apart in the genome and still have pretty good efficacy. So there's a paper from George Church's lab that shows that you can get end-join mutagenesis and homology-dependent repair using these double nicking systems. This is a paper from Feng Zhang's lab showing essentially the same thing. And one of the things they've done is that they've shown that with the double nicking, that your specificity is better, that the ratio of mutagenesis at defined off-targets the on-target to the defined off-targets gets better with the double nicking, that's in the shaded bars here, than it is with the double-strand break-inducing wild-type Cas9 protein. And this is just showing that you can go some distance apart. They looked at the separation between these. You can go some distance apart and still get pretty good efficacy. I should emphasize that single nicks are not very effective. Nancy Maisels at the University of Washington has shown that you can get some efficacy out of a single nick, but it's not as good as double nicking. This is looking at homologous repair. I guess all of these are looking at homologous repair. And when you're doing double nicking, generating a 5' overhang, so you have a nick on one strand, nick on the other strand, if it's 5' ends that are overhanging between the nicks, that works better for homologous repair than the other way around. And I can tell you why I think that is if you ask me. Okay. Another thing that Jinsoo Kim found was that if you just put a couple of extra Gs on the 5' end of the guide RNA that don't match the target, it actually improves the specificity a little bit. And this is just data from his paper, and I can tell you in a minute why I think that works. And Keith Jung's lab said, well, what if we shorten the length of the guide sequence? We just take a single guide RNA, instead of making the match to the genome 20 base pairs, we make it 19 or 18 or 17. And it turns out for many of these guide RNAs, going to 17 or 18 base pairs doesn't reduce the indel frequency, but it can still support homologous repair, but it does improve the specificity. Okay? And this is showing for single base mismatches, full-length guide RNAs versus truncated guide RNAs, and this is for two-base mismatches. And you can see that whereas this guide RNA actually isn't very sensitive to single base mismatches even when you use a shorter one, but this one is very much more sensitive to single base mismatches, and this one is quite a bit more sensitive. And even this one is much more sensitive to two-base mismatches. So they also went and they did guide-seq Whereas initially at this particular target, the design target was third best and represented 11% of the reads. Now with a 17 base guide RNA, it's moved into first place and represents 30% of the reads. So it's improving things. I had talked a little bit about donors that take advantage of the displaced strand. Jennifer talked about that a little bit. Ian Slaymaker in Feng Zhang's lab said, well, part of the energy of binding of Cas9 and the guide RNA to the target comes through nonspecific ionic interactions between positive charges on the protein and negative charges on this displaced strand. So if there's excess energy at the design target, that means that secondary sites that are only partially matched now have enough energy to get bound and cut. What if we reduce the energy of interaction to the displaced strand? And so there was no crystal structure at that time that actually showed where the displaced strand is. Now there is one, or at least that cryo-EM structure. But they thought they could identify positively charged residues out here that were good candidates based on the existing crystal structures. And so they mutated them one at a time and looked to see what's the efficiency of cleavage at the design target and a couple of identified secondary targets. And they found ones that seemed to cut and make indels in the design target quite efficiently, about as efficiently as the wild type, but significantly reduced the cleavage at these secondary sites. And they then began combining these mutations. They're substitutions of alanine, uncharged amino acid, for positively charged lysines or arginines. And they eventually came up with enhanced Streptococcus pyogenes Cas9 1.1 that has 3 of these substitutions and shows very high cleavage at the design target and very little at the secondary sites. So that's a way to enhance specificity. And this is just showing the specificity. Here's 1.1 out here that is, and also here it's quite sensitive. Remember, blue indicates insensitivity, white indicates sensitivity. It's much more sensitive to mismatches than the wild type, and to single mismatches, and very sensitive to these double mismatches. Now, Keith Jung's lab had the same idea, but they went for the other strand of the target. They went for the RNA-DNA hybrid, and they actually had a crystal structure that showed the contacts between the protein and the DNA-RNA hybrid. And so they identified some residues they thought they could mutate that would reduce the binding energy between Cas9 guide RNA and the target, and they made mutations in those. And similarly to what the Zhang lab had shown, they found increased specificity by combining a number of these mutations. These are, again, alanine substitutions. This is arginine, asparagine, and glutamine in this particular case. And they did guide-seq on these. They didn't actually, in this paper, do guide-seq on the same target I showed you earlier, but they did guide-seq on a bunch of other targets and showed that, here's one where the wild type showed pretty good cleavage at the design target, but lots of capture of sequences elsewhere in the genome. And this high fidelity 1 version of Cas9 protein picked up no sequences from these other sites using guide-seq. And there are a bunch of them where the specificity was very greatly enhanced. So you've got all of these methods to improve specificity. I think that the truncated guides, the and I think it's also true of these 5' GG extensions and these mutations in basic or polar residues in the protein may all work by eliminating that excess affinity. So if you bind the target more tightly than you absolutely need to, you've got this excess affinity that means that the secondary targets, although they have some mismatches that reduce affinity, they've still got that extra affinity from other interactions. And if that's true, then the on-target affinity is going to depend on the sequence of the target and the guide sequence. So for very AT-rich targets, you may not be able to employ some of these affinity-reducing measures. You'd be better off going with wild type. But if you've got a very GC-rich target, where the RNA-DNA hybrid energy is still really high, you may want to employ some of these other approaches. As far as I've seen, people who are doing sort of routine use of the system have not started to employ these high-fidelity modifications. They're still kind of going with the basic system. But as they get more finicky, they may need to. It's also true, as I think I said before, that the CPF1 homologs seem to have higher inherent specificity. So that's another thing you can do. And this is just showing, this is comparing, well, there are a variety of things going on here. This is from Jinsoo Kim's lab. But one of the things it's showing is that the off-target cleavage can be very much lower. I'm looking for one where they actually had, well, the comparison that they have done with Pyogenes Cas9 has to do with doing a lot of sequencing of the cut sites and making a sequence logo for, what are the preferences of the CPF1s and Cas9. And Cas9 obviously can tolerate some substitutions, particularly far from the PAM, whereas the CPF1s, again, far from the PAM, they tolerate more, but these are much more well-defined sequence logos indicating higher specificity. This was an interesting observation. They set up a situation where these two CPF1s from two different bacteria and SP-Cas9 actually cut the same sequence, just based on the disposition of the PAMs. And what they found was that even between the two CPF1s, the indel signature was somewhat different. This CPF1 preferred making a 15-base deletion with a minus 6-base and a 3-base deletion, whereas this one, cutting the same target, preferred the 6-base deletion and had a 16-base deletion as the secondary preference and little or none of the 3-base deletion. So there's something about the proteins, not just the underlying sequence, but something about the proteins that's influencing the indel outcome. And then the Pyogenes Cas9 cutting in the same site had different preference. And as I had said before, if you do the experiment multiple times, you get the same preference for the same protein and guide RNA. Okay. And this is GuideSeq looking at the CPF1 homologs. And again, you're seeing situations where they're getting very little capture of breaks at secondary sites in the genome. One of the CPF1s gives a few other sequences. This one's giving essentially none. And the reason that there's just a single line in many of these is they'd captured nothing but the design target with CPF1 cleavage. So if you're worried about specificity, then you have things you can do about it. First, you can design a new guide RNA. That's what people tend to do initially. You can use paired nicking. You can use this mismatch GG extension. You can make the truncated guides, the We actually did that when I was here working at IGI. We made I think it was a 17 or 18 base guide sequence that worked very well. You can use these Cas9 mutants from the Zheng lab or the Zhang lab, CPF1, and I'm going to mention on Wednesday why using RNP delivery has some advantages for specificity over some of the other delivery methods. But there are a lot of things you can do to try to enhance the specificity. And since this lecture was called editing issues, I'm going to do something like what Jennifer did very briefly. I'm going to emphasize a couple of different things from what she talked about. She was talking about the applications that themselves and how they would influence society. And what I'm going to do is I'm going to talk about two issues that I think are quite important, and these may come up again on Friday, I don't know. One is whether you're talking about making changes, enhancements of agricultural organisms, or changes in humans to fix them, you have to decide at the outset who needs to be fixed. What are you going to change? What people are you going to tell need to be fixed because they aren't right? Okay? And then once you decided what to fix, how are you going to distribute that fix to the organisms, people that need them most? And even in the realm of agriculture, I showed you an example of modifications in wheat. I showed you an example of dehorning cattle. Well, what other changes should be made? And are they changes that would benefit farmers in Wisconsin, or are they changes that should benefit farmers in sub-Saharan Africa? Okay? So on the human side, some of you may know this book by Andrew Solomon. It's called Far from the Tree, and it talks about people who are unusual in various ways. And one of the groups he talks about are people of short stature, have genetic causes for being very short. And he makes the point that many people who have are born with short stature wouldn't change that, even if they could. They're comfortable with who they are, and they in many cases wouldn't even want a genetic change that would make their children tall instead of short. So you have to be careful who you identify as needing to be fixed. Okay? In the realm of distribution, when I was here on sabbatical 2 years ago, we worked on a project to fix the mutation in sickle cell disease. Well, here we are. We're sitting up here in Northern California, and there are sickle cell patients in Oakland, maybe a few in Berkeley, but the vast majority of sickle cell trait carriers and disease sufferers are in tropical regions in Africa and Asia. And if we develop this fancy, expensive molecular technology, how do we get it to the people, the huge numbers of people who really need it? So these are issues that come up sort of post-technology. What are you going to do with these tools that you now have? So I just wanted to highlight that, and that I didn't want to show. So questions about Cas9 variants and off-target issues? Yes?
The off-targets you mentioned, if I remember right, for crop modification, maybe if you start from a single crop at the end, would be whole genome sequencing would be okay. Otherwise, you wouldn't say people should use it, or?
No, I think so. Particularly for the agricultural organisms, plants and animals, the phenotype is the key, right? You want to test the product and not care as much about the method by which the product was generated. So you can do whole genome sequencing. There's a disadvantage to doing whole genome sequencing. There's no chance that the genome sequence of one of those bulls I showed you earlier is identical to his parent, okay? It's just not going to be true because of the number of cell divisions and the mutations that accumulate just during cell proliferation. So there are going to be differences. And if you insist that there be no differences, you'll never do anything. So whole genome sequencing can reveal things that are pretty common and look like things that could have been generated by off-target cleavage, but you have to be careful about what you do. I don't know, how many people noticed this paper that appeared in Nature Methods a little while ago that said, unanticipated high levels of off-target mutagenesis. You should ignore that paper absolutely completely. Maybe those of you who haven't seen it, it's in Nature Methods, June maybe, May or June. Very recent. The first author is Schaefer, so if you want to look it up, I can give you the reference to it later if you're interested. And maybe we should have a quiz later. So people need to tell me why you should ignore this paper. What is it? What's wrong with this paper? So anyway, you should ignore it. It's nothing it says is true. Yeah.
I was just wondering, with like the other Cas9 variants, if you look at the CRISPR arrays in the species, do you see like that the guide sequence is longer or shorter? Like, was that kind of like the origin of adjusting the length of the guide sequence, or did they just kind of brute force that?
Did who brute force it?
The people who studied the variants. Did they be like, well, I'm going to try to get 1921, just sort of sweep that?
So the CRISPR RNAs are developed from a long transcript. And when that transcript is processed, you don't get the whole of what we call spacer in the final CRISPR RNA. So you can't just look at the size of the spacers and from first principles decide how much is going to appear in the ultimate CRISPR RNA. So they could have gone and looked at the natural CRISPR RNAs, but it was easier to look at this in more of a technological setting. It's also true that the repeat sequence, part of that is eliminated during processing.
So because we know our sequence of our guide RNA, theoretically, bioinformatically, you should be able to predict what the off-target sequences would be. Do we find that to be the case when we compare it to the empirical results of, say, GuideSeq?
There's an imperfect overlap between predicted and detected secondary targets. And I don't know that anybody really knows the reason for that. I don't know. I guess it's on Wednesday that Jennifer Listgarten is going to be here, and she's developed some tools for predicting good targets. And you could maybe ask her that question. But it is surprising that sometimes a single mismatch doesn't appear as frequently in the GuideSeq study or one of these other studies as a 2 or 3 base pair mismatch. And I went and I tried to do something just trying to use energetics to predict what targets would be more susceptible to mismatches than others. There was an imperfect overlap with that and experiment also. So I don't think anybody really has a great way of rationalizing that.